Hello all, myself Dr. Pooja. I welcome you to the second lecture on capsule staining. In previous lecture we have seen Manuel's method, which was an example of negative capsule staining technique. In today's lecture we will see his method. Now this method which we are going to see in today's lecture is modified by Anthony. Originally it was a method of his, but it was modified by Anthony. So let us begin with steps that we will be carrying out for performing this his method. We will take a drop of crystal violet on a clean grease-free slide, to which a looful of bacterial culture will be added. Then using a second slide we will prepare a thin film as we have prepared in negative staining. Then 20% copper sulfate solution will be used as a decolorizer. We will wash that slide with copper sulfate solution. This is what an observation of Anthony's method or his method, where we will observe a colorless capsule against a purple colored background, and again bacterial cell will be stained with dark purple color, which is basically due to crystal violet solution. So in this method we will use two reagents. First one is crystal violet and second one is a 20% copper sulfate solution. Now let us see introductory part. Actually we have already seen this part in our previous method of manuals. So in this lecture we will quickly go through this introductory part. Now what is capsule? As we know, it is a gelatinous outer layer which is present outside this cell wall, and it protects that bacterial cell. Now those bacteria which are able to produce capsule, they are observed as virulent. Virulent means they are capable of causing diseases. As a result, they are not easily killed or they are not easily attacked by the host immune system. So that capsule protect bacterial cell against phagocytosis, and this capsule makes staining difficult. So they are not easily stained by some differential staining techniques, because capsule is soluble in water, and if we used water for washing that smear, that capsule can be easily washed away. So that's why a special staining techniques, special stains are required for staining capsule. And one of the most important thing that to be kept in mind while performing capsule staining is smears should not be heat fixed, because heating that smear will be responsible for destruction of capsule, as it is a delicate structure, and that will also cause a shrinkage, and as a result it might produce some kind of artifact which is mistaken for capsule. त्यामुळे कॅप्सूल स्टेनिंगमध्ये मग ती हीच मेथड असो किंवा मॅन्युअल्स मेथड असो, हीट फिक्सेशन स्मिअरचं करायचं नाही. आणि जेव्हा वॉटरनी वॉश केले जाते मॅन्युअल स्टेनिंगमध्ये, तेव्हा ते जेंटली वॉश केलं जातं स्मिअर. हीच मेथडमध्ये कॉपर सल्फेट सोल्युशन इज यूज्ड ॲज अ डिकलरायझर इन्स्टेड ऑफ वॉटर. Now let us see history. Capsules, as they are difficult to stain, they require a special staining techniques. Now they can be visualized by using electron microscopy, by using phase contrast microscopy, and some staining techniques. Those can be broadly classified into positive staining technique for capsule and negative staining methods for capsule staining. Example of negative staining technique which we already discussed in our previous lecture was dry India ink film and method of Manuel. So we have seen this Manuel's method, and we have already seen this India ink method. Now in today's lecture we will talk about his method, which is modified by Anthony. So apart from this his method, other examples of positive staining methods are method of White and method of Molar. Now let us see his method. So this his method, it was modified by E. Anthony in 1931, and that's why it is also called as Anthony's capsule staining method. Primary stains. Now this his method uses two stains. Two reagents are used in his method. First one is crystal violet and second one is a 20% copper sulfate solution. So the crystal violet is used as a primary stain and 1% of crystal violet is used. Now this crystal violet, when applied to a non-fixed smear, here the bacterial cell and capsule will be stained as a purple colored. But as capsule is not easily stained, the only bacterial cell or cell wall will take this violet stain and it will be observed as violet colored, purple colored. On the contrary, although the stain adheres on capsule, it is not able to bind. कशामुळे? कारण कॅप्सूल नॉन आयनिक असते आणि जरी त्यावर स्टेन पार्टिकल्स ॲडहेर झाले असले तरी ते बाइंड होत नाहीत, त्यामुळे ते इझिली डिकलराइज होऊ शकते. आता दुसरं रिएजंट काय आहे हीच मेथडमध्ये? तर 20% कॉपर सल्फेट सोल्युशन. Now capsule, as stated earlier, it is non-ionic and it is not easily stained by primary stain. So it is that primary stain is not able to bind to capsule. Hence, when copper sulfate is used as a decolorizing agent, it washes that purple colored primary stain from that capsule, but it is not able to remove stain that is bound to cell wall. सेल वॉलवर जो काही क्रिस्टल वॉलेट बाइंड झालेला आहे ते कॉपर सल्फेट काढू शकत नाही. तो फक्त कुठला स्टेन काढेल? जो काही कॅप्सूलवर ॲडेअर होता तितकाच फक्त रिमूव्ह होईल या कॉपर सल्फेट सोल्युशननी. कॉपर सल्फेट इथे काय म्हणून वापरलं? डिकलरायझर म्हणून वापरलं. त्यामुळे काय होईल? आता सेल वॉलनी कुठला स्टेन घेतला? प्रायमरी स्टेन. प्रायमरी स्टेन कोणता यूज केला आपण? क्रिस्टल वॉलेट. ॲज अ रिझल्ट, cell wall or bacterial cell will be observed as a purple colored, violet colored, whereas this capsule which is decolorized by copper sulfate solution, it will be visualized either as a colorless or in light blue color, and background is also stained by crystal violet, so it will also be observed as a purple color. Now let us see requirements. The requirements will be bacterial suspension of capsulated microorganisms. In previous lecture we have seen examples of bacteria which are able to produce capsule. कुठले कुठले examples होते? सर्वच बॅक्टेरिया की सर्वच मायक्रोऑर्गॅनिझम कॅप्सूल तयार करतात का? नाही. काही स्पेसिफिक असतात जे कॅप्सूल प्रोड्यूस करू शकतात, त्यांना म्हणतात कॅप्सुलेटेड बॅक्टेरिया और इनकॅप्सुलेटेड बॅक्टेरिया. काही ग्राम निगेटिव्ह बॅक्टेरिया जसे की इश्चेरिशिया कोलाई, ग्राम पॉझिटिव्ह बॅक्टेरिया जसे की बॅसिलस अँथ्रासिस हे आहेत जे कॅप्सूल प्रोड्यूस करू शकतात. So first requirement will be bacterial suspension. Second will be reagents. Which two reagents? 1% crystal violet and 20% copper sulfate solution. And some general requirements, some general materials such as inoculating needle, glass slide, staining tray, staining rack, disposable gloves, and microscope to visualize our slide. Now let us see procedure of his method of capsule staining. Take a clean grease-free slide. Prepare a smear on a clean grease-free slide. Allow it to air dry, but do not heat fix that smear. Then cover that slide with 1% crystal violet. Allow it to react for 2 minutes. And then, instead of water, use copper sulfate solution for rinsing that smear. Allow it to air dry and by putting a drop of oil immersion, observe it under oil immersion lens. काय करायचं? एक क्लीन ग्रीस फ्री स्लाइड घ्यायची, त्यावर स्मिअर प्रिपेअर करायचा, त्यावर 1% क्रिस्टल वॉलेट टाकायचा, नंतर काय करायचं? त्याला 2 ते 3 मिनिट रिॲक्ट होऊ द्यायचं आणि 20% कॉपर सल्फेट सोल्युशन वापरून त्या स्लाइडला वॉश करायचं. जेंटली वॉश करायचं, कारण कॅप्सूलला आपण काय केलं नाही? हीट फिक्स केलेलं नाही आहे. हीट फिक्स का करायचं नाही? बिकॉज हीट फिक्सेशन विल बी रिस्पॉन्सिबल फॉर डिस्ट्रॉईंग कॅप्सूल. इट विल डिस्टॉर्ट द कॅप्सूल और इट माइट कॉज श्रिंकेज ऑफ सेल. त्यामुळे हीट फिक्सेशन करायचं नाही. त्यामुळे स्मिअरला जेव्हा आपण वॉश करतो या कॉपर सल्फेट सोल्युशननी, तेव्हा ते जेंटली ड्रॉप बाय ड्रॉप वॉश करायचं. सो आफ्टर दॅट काय करणार आपण? स्लाइड एअर ड्राय करायची, त्यावर ऑइल इमर्शन टाकायचं आणि ऑइल इमर्शन ऑब्जेक्टिव्हमध्ये बघायचं. त्याच स्टेनिंगला आपण दुसऱ्या पद्धतीनेही करू शकतो. ते कसं? तर एक क्लीन ग्रीस फ्री स्लाइड घ्यायची, यावर क्रिस्टल वॉलेटचा ड्रॉप पहिले टाकायचा, 1% क्रिस्टल वॉलेट टाकायचं, त्यानंतर लूपफुल ऑफ बॅक्टेरियल कल्चर घ्यायचं. लूपफुल ऑफ बॅक्टेरियल कल्चर घेऊन दुसरी एक स्लाइड घ्यायची. दुसऱ्या स्लाइडनी ज्याप्रमाणे आपण निगेटिव्ह स्टेनिंगमध्ये थिन फिल्म तयार केली होती त्याप्रमाणे याची थिन फिल्म तयार करायची आणि नंतर याला 20% कॉपर सल्फेट सोल्युशनने वॉश करायचं. आणि मग जेव्हा ती एअर ड्राय होईल त्यावर ऑइल इमर्शनचा ड्रॉप टाकायचा आणि ऑइल इमर्शन ऑब्जेक्टिव्हमध्ये ऑब्झर्व करायचं. या कुठल्याही प्रकारे आपण ही कॅप्सूल स्टेनिंग मेथड बाहीस करू शकतो. आता काय ऑब्झर्वेशन मिळेल आपल्याला या मेथडमध्ये? मेथड ऑफ हिस किंवा अँथनीज मेथडचं काय ऑब्झर्वेशन असतं? आपल्याला यामध्ये लाईट ब्ल्यू कलरचा किंवा मोस्टली कलरलेस और ट्रान्सपरंट कॅप्सूल विल बी ऑब्झर्वड. बॅकग्राऊंड जो आहे तो पर्पल कलरचा आहे. अँड अगेन बॅक्टेरियल सेल विल बी स्टेन्ड ॲज अ डार्क पर्पल because of application of crystal violet, which is a basic stain, and it is taken up by the wall of bacteria. As a result, it will be observed as a purple color. This was all about method of His, which was an example of positive capsule staining method. Hope you have understood the concept. In case you have any kind of difficulty, you can ask that in comment section. Thank you. Soon we will meet with another lecture where we will see pH staining.